Welcome to your review on blood. My name is Lee Dornty. Blood is a connective tissue. You wouldn't inherently think of it as a connective tissue, since it doesn't exhibit the properties of other connective tissues such as cartilage and bone and the structure formation. But it is because of the formation of the plasma matrix. Blood has four major components. Plasma, which is the matrix. It is the clear solution that the blood is suspended in, the red blood cells are suspended in. It's predominantly just a saltwater solution with some proteins. Erythrocytes, which are red blood cells, that's your oxygen carrying capacity. Leukocytes, which are your white blood cells, which are your body's natural defense against invaders. And platelets, which are actually little formations of cells. This is your clotting factors. So how much blood does a human have? An adult human has between 4.5 and 5.5 liters. That's about 1 to 1.5 gallons of imperial. The components of blood are typically measured in hematocrit. Hematocrit is a way of determining the percentage of each of the elements of blood. Plasma will be one section. The buffy coat, or better known as just formed proteins or formed elements, will be the central coat. And then the bottom section, which is the actual hematocrit percentage, is your red blood cells. Sometimes you'll see hematocrit also labeled as red blood cells and formed elements together, but effectively this makes almost no difference to the actual percentage, as typically it only adds decimals of a point. The way hematocrit is done is you simply prick your finger, take a few drops of blood into a capillary tube, spin it in a centrifuge, and then measure the blood out. What you're looking for is what percent is the red blood cells over the total blood. Normal blood for females should be about 37 to 47%. For males, it's a little higher. It's typically 42 to 52%. When you're below that, it's considered anemia, and when you're above that, it's considered polycythemia. There are factors that can affect your hematocrit. Most of them are artificial, some of them are not. One of the things that's common is EPO, or erythropoietin. EPO itself is a hormone that your body naturally makes that tells your bone marrow, we need more red blood cells, please produce them. This can also be abused by simply injecting it. One of the great examples made famous by our yellow bicyclist, he used to do EPO and blood doping. While EPO tells your body to just make more red blood cells, blood doping is where you will take your actual blood out, store it, and then place it back later after your body replenishes those red blood cells. Why would anyone do this? Well, quite simply, your red blood cells is a physical measurement of how much oxygen you can process. While things such as the metabolic cart will tell you how well you process oxygen currently, meaning what is your max because of your fitness level, your red blood cells are one of the indicators of what your max potential is. If you raise the amount of red blood cells you have, you can carry more oxygen. You can carry more oxygen, you're a better athlete, you have more endurance. Why would you not want this? Simple. When your red blood cell level gets too high and your hematocrit level gets too high, your blood starts to turn into syrup. It becomes very, very thick. This causes quite a lot of medical problems. Stroke is one of the common issues that happens when your blood is too thick. On top of that, there's quite a lot of other issues. One of them being that your cardiac cycle is working far too hard. When your blood is that thick, it is pumping furiously just to be able to move it. And it's causing a lot of back pressure against your heart. It's not circulating well. This is going to cause a lot of issues. Cardio hypertrophy being one of them. Your left ventricle will enlarge, and this can be fatal. This can cause issues such as backing up the blood into the lungs. You can quite literally drown on your own blood. It can cause aneurysms. It can cause different ruptures, bleeds, all kind of issues can happen. And eventually, when your heart works that hard, pushing that heavy of a load, it's going to give out. So eventually, it will cause cardiac arrest as well. Erythrocytes are your red blood cells. Red blood cells are the most common type of blood cell you have, and they're the principal means of delivering oxygen to your body tissues. Red blood cells take up oxygen in your lungs and then release it to your tissues while they squeeze through the body's capillaries. The cytoplasm of an erythrocyte is rich in hemoglobin. This is an iron-containing biomolecule that can bind oxygen and is responsible for the red color of the cells in the blood. Each human red blood cell contains approximately 270 million of the hemoglobin molecules. The cell membrane is composed of proteins and lipids, and this structure provides properties essential for physiological cell function, such as deformity and stability. Basically, so it can squish and get through your blood vessels easier. In humans, mature red blood cells are flexible and they're oval biconcave discs. They lack a cell nucleus and most of the organelles in order to accommodate a maximum space for hemoglobin. Approximately 2.4 million new erythrocytes are produced per second in human adults. The cells develop in your bone marrow and they circulate for about 100 to 120 days in the body before their components are recycled by macrophages. Each circulation takes about 60 seconds. Approximately 84% of the cells in the human body are actually red blood cells. Nearly half of the blood's volume is red blood cells. Leukocytes are white blood cells. There's 5 types: neutrophils, lymphocyte, monocyte, eosinophil, and basophil. Neutrophils make up about 60% of the white blood cells. You will see them the most active and the most plentiful when there is a bacteria or a fungus invasion. You can spot a neutrophil under the microscope by the 3 lobules or the 3 or more lobules. Sometimes they'll have more. They look a little more distinct. The one they're most commonly confused with is eosinophil, but eosinophil, if you'll notice, only has 2 lobules. Lymphocytes make up about 40% of your white blood cells. They're most prominent in the detection of viruses. Lymphocytes are most notable under a microscope because they take up almost the whole lymphocyte with its central core. It looks like one giant purple dot. Makes it much easier to see than all the other ones. Monocytes make up about 8% of the white blood cells. They're most prominent during chronic infections. They also have the one single core, much like lymphocytes, but it's typically in a U shape. Eosinophils make up approximately 2% of the white blood cells. They're most prominent during parasites. They're most easy to recognize under a microscope because of their 2 lobules, and sometimes there's also a little dot and people like to think that it looks like a smiley face. Basophils are very rare in your white blood cells. They're not actually 0%, but you're at such a small percentage that it's basically none. They're most prominent during allergies. They're a histamine reaction. They're easy to identify if you can find one because they're very granulated. If you look at it, it looks like it's completely covered with sand. Nice little mnemonic for remembering what they are is 60-40-820, never let my engine blow. This is to tell you that 60% are neutrophils, 40% are lymphocytes, 8% are monocytes, 2% are eosinophils, and about 0% are basophils. Blood pH. The pH of your blood should be between 7.35 and 7.45. Anything outside of this is bad. Now below 7.35 is acidosis, and above 7.5 is alkalosis. One of the questions I remember seeing on one of my medical exams that confuses a lot of people is, what is a pH of 7.25? It's acidosis. This can be confusing because above 7 is typically considered alkaline, and below 7 is considered acid. However, when you're talking about blood pH, 7.35 and 7.45 are your normal neutral markers, not 7. So you just have to remember anything below 7.35, acidosis. Anything above 7.45, alkalosis. Generally in medicine, we consider 1 point to be detrimental and pretty much going towards death. So below 7.25 and above 7.55 is quite dangerous. Which brings to the question, what about all these things such as alkaline waters that say they have a pH of 9 and they'll make your body alkalotic and it's supposed to be quite good for you? Is it true? No, absolutely not. It does not do anything. Water is water. If it did bring you down, it would be quite detrimental. So what is one substance that would make your blood more alkalotic? Well, bleach will work, but as we all know, if you actually was to take bleach and your blood really was to become alkalosis, it would become instant death. So, the water that says it'll bring your blood to 9.0 and make you very alkalosis, luckily, it's all just a bunch of crap because otherwise it would kill you instantly. Blood typing. First, what is it? Blood typing is just a way to see what blood is going to be compatible with which person. There's considered to be 4 types of blood, but in truth, there's only 2 protein markers that really make your blood. You either have A, you have B, you have both A and B, or you have O, and O is simply no A and no B. So how do we actually identify blood? Well, we have things called antigens and antibodies. The simple version to remember it, antigen is a way of saying this is self, this belongs here. Or when you have antibodies, antibodies go, this is not self. And if this is not self, it wants to kill not self. So in your blood, you have your red blood cells. And you have your antigens. If you're type A, that means you have A antigens. The blood recognizes self as A. If you're type B, you have B antigens. The blood recognizes self as B. If you're A and B, you have both. You recognize A as self and you recognize B as self. If you're O, you have neither A or B. Therefore, you recognize no self. The flip side of that is antibodies. Antibodies look for what you're not. So, if you're A, you have A antigen, but you have anti-B antibodies because it does not recognize B. So you'll have an A antigen, but an anti-B antibody. This is a point that commonly confuses a lot of students. Flip side with B. If you're type B blood, you have B antigen, but you have anti-A antibodies, which means you will attack A's. If you have A and B antigen, you recognize both as self. Therefore, you have no antibodies. If you have neither A or B antigens, then you're going to make both anti-A and anti-B antibodies. So when you're looking at this blood, if you have A and B antigen, you can accept anybody. That's why AB is the universal recipient. If you have type O, you can only accept other O's because you're making antibodies against A and B, but you have no antigens. So you can put type O blood in anyone, and there is no self markers to recognize, therefore nothing to attack. So how do we blood type this? Well, we simply prick your finger, get a slide, put 3 drops of blood on there, put the antibodies on, and see if it reacts. Besides A and B, there is one more protein, and that's the Rh factor. The Rh factor is what gives you that plus and minus. Rh factor is simply an abbreviation for the first way we discovered it, which is rhesus monkey. So, you have a drop of blood, such as on the slide here. You've put it in 3 columns. You dump your anti-A on one, your anti-B on one, and your anti-Rh on one. Well, that A has had a reaction. That blood has been destroyed. Because it's been destroyed, we know there's A antigens there because we put antibodies on it. B, no reaction happened. Rh, that blood's been destroyed, so we know it's positive for that Rh. So A's destroyed, B's fine, RH is destroyed. So that is A positive blood. The next column, it's a little misleading because that sample is so small. They put antibodies on A, and there was no reaction. That's just the amount of blood they had. They put anti-B on B. That blood's obviously been obliterated, so that's a positive test. And they put anti-RH on the factor, and that blood was obliterated, so that's also a positive test. So this blood is going to be B positive. On the third column, you can see in the anti-A column, A was destroyed, so that's positive. B was destroyed, that's positive. RH was destroyed. So yes A, yes B, yes positive. The third column, A is fine, B is fine, and O is fine. The fourth column, A is fine, B is fine, and the RH factor is fine. So no, it's not A, no, it's not B, no, it's not positive. If you're not A, you're not B, you must be O. And if you're not positive, you're negative. Good luck on your exams. Hope this helped.